Use 1/3 of distillated water. Next, add 1 tablespoon of dish soap. Lastly, add half a teaspoon. This creates your buffer. I will be comparing strawberries, blueberries, and cherries. We go ahead and add 50 milliliters of our buffer solution into our strawberries. We then begin to mash our strawberries with the buffer solution in order to break the cell membrane. We continue to do this until we are satisfied with our outcome. Once we are finished mashing, we will go ahead and pour it out into our filter paper. After pouring it into our filter paper, we will release all the liquid, as much liquid as we can, in order to get every piece of DNA out. Now I am adding 91% alcohol into my solution. Add as you find pleased. Here the DNA is shown. I am going to go ahead and collect it into this jar. We will now repeat the process with our cherries. Go ahead and add 50 milliliters of our buffer solution into it. Then mash until you are pleased with the outcome. We now go ahead and pour our cherry solution into the filter paper. We then filter out any of the liquid. I then again use the 91% alcohol and pour until I am pleased. Here, very few strands of DNA are shown. With our blueberries, I only went ahead and added 25 milliliters of our buffer solution since they are smaller fruits. After doing so, mash until you are pleased with your final outcome. We then again filter out our liquid until you have all the liquid that you find necessary. I went ahead again and used the 91% alcohol and poured until I was pleased. Barely, if any, DNA was shown. I collected the samples that I had seen and barely anything was seen in the liquid. Here I am showing each of the DNA samples.